Hello, everyone. I'll talk about SHIP, an annotation-based program for identify genomic safe harbors in eukaryotic model organisms. So since the beginning of genetic engineering, the insertion of exogenous genes into host cells has presented itself as an important challenge. With time and the advent of synthetic biology, the challenge became even greater with the expansion of monogenic to polygenic traits of interest and the need to insert metabolic pathways and entire genetic circuits. So whatever the insertion of new gene models must not interfere with existing cell genes or interfere with their expression. In other words, the insertion space is constrained and should be carefully selected. Such regions are classified as Genomic Safe Harbours, GSHs. And what is a genomic safe harbor? I really like this definition made by Salan et al. That GSHs are regions of the genome that we can put or insert without adverse effects on the host cells. We have two examples of some of the most used GSHs in humans, and some concerns are that they are in intragenic regions with high genetic density and close to oncogenes, these head boxes. And there are some GSHs described in other organisms with similar concerns, and it becomes clear the need for tools allowing the optimized identification of these regions. We propose to identify GSHs in intergenic regions following defined criteria that are of the great importance for many studies carried out in cell cultures, in animals, for the biotechnology industry, and who knows in the future with using gene therapy. So our work follows the DBTL cycle, which concerns of 4 phases: the design, build, test, learn, and repeat if something gets wrong. We are in the first one. Our strategy to identify GSH was to develop the SHIP solution that brings an optimized approach, with the input being a GFF file, and the output of this will be a genomic overview, a list of pGSHs, the putative genomic safe harbors, and their regulatory features. And this part can be used for any organism with a GFF file. For this project, we chose 3 model organisms to be evaluated: the yeast, Saccharomyces cerevisiae, mouse, and human, for having a complete genomic sequence and well annotation. Now I will present the results at applying to the program. So here we have the identified 6 pGSHs in Saccharomyces cerevisiae. We have 10 pGSHs in musculus and 16 pGSHs in humans. So we use machine learning to ensure that our pGSHs do not have other sequence that interfere with genome expression, and the organism chose to do this was the human, as it has a precise annotation of the genome. We developed a 3-class identification model with a high accuracy in the training phase, and was able to identify all classes in the test sample that we used to assess if the program was really working and not overfitting. And then we use it to run the actual pGSHs that we identify, and all were predicted as contain code and sequence. For this preliminary result, we have some hypotheses that will be detailed in another occasion. In conclusion, unlike the empirical methods used in previous studies, the SHIP provides a more efficient analysis in identifying pGSH, enabling a refinement and systematization of this search. About the machine learning, we need to improve the approach and reevaluate the choice of analyzed features, but the analysis performed provide an initial effort to reduce possible GSHs to be validated with experimental data. Next, we will start in the in vivo validation with Saccharomyces cerevisiae. This is our team. You can find us at this email. My name is Matheus Leitão, and this was SHIP. Bye-bye.